Hello students, today we are going to discuss about another video tutorial upon cell biology. So we are going to discuss today about DNA replication. Before discussing about DNA replication, we are going to discuss about cell, then discuss about DNA which is present in the nucleus, and discuss about detail about the DNA replication. So let's start. Suppose it is a cell and it contains a central core that is called nucleus, and this nucleus contains a structure, wrapped structure, which is wrapped with a type of protein is called histone protein. Histone protein is wrapped with its nucleic acid, means it wrapped with the chromosomes, and the chromosome contain the DNA. So if we open this content from the nucleus, this is suppose a chromosome long stretch structure. Suppose this is a chromosome and its small part is called DNA. And the DNA contains small subunit, and these are called genes. They are called genes. So they are genes. So we are going to discuss about DNA replication very briefly. So come to the point. So a cell contains a nucleus and it contains a chromosome. This is called a chromosome. When it starts to replicate, in two ways. In case of prokaryote, it starts at a specific point. Suppose in prokaryotic cell, it is DNA and start at specific site. This is called ORA replication. This is a specific site in which the DNA replication is initiated, and the DNA is open up to this direction. It is a specific direction. But in case of eukaryotic DNA, the chromosome is going to start to replicate at different points. The DNA replication is start at different points. Suppose here. There is several points of origin of DNA replication. In prokaryote, it is at a specific point, but in case of eukaryote, it starts at several points. Suppose I am indicating the points. Suppose A, B, C, D. Suppose here this is indicated as M. So in prokaryote, the origin of DNA replication is M, and the DNA replication is a unidirectional. It is specific direction. The DNA replication start at a specific point and going to a specific direction in case of prokaryote, but in case of eukaryote, it start at several points. Suppose in A, here B, C, D, there is many points is there where DNA replication is going to be continued. And that's why in eukaryote DNA replication start at several points. If we observe the DNA structure, the DNA is made of three major components, made with three major components. One is nucleic acid, one is sugar, nitrogenous base, and phosphate group. Phosphate group. So the DNA is made with three important components basically. One is sugar. In case of DNA, it is deoxyribose sugar. In case of RNA, it is ribose sugar. Sugar is two types. So in DNA it is deoxyribose sugar, and in RNA it is ribose sugar. Now the nitrogenous base. So what are the nitrogenous base? These are actually four types. In case of DNA, and in case of RNA, it is the thymine, which is replaced by uracil. So the nitrogenous base are again I am drawing here. In case of DNA, nitrogenous base are adenine, guanine, thymine, another is cytosine. Adenine, guanine, thymine, cytosine. In case of DNA, there is four types of nitrogenous base. In case of DNA, in case of RNA, it is adenine, uracil, cytosine, and guanine. And the phosphate group is phosphoric acid. So now we are going to discuss how the DNA replication actually happens and what are the enzymes which are mainly involved in this process. So the enzyme in DNA replication. So in DNA replication there is many types of enzyme is present. One is topoisomerase. Topoisomerase. This is the types of enzyme which are helps in DNA replication. This is the first enzyme which are going to initiate the DNA replication. So which is present in the one side of the DNA replication, and it's going to initiate the DNA replication. Topoisomerase is followed by another enzyme. This is called helicase, which is play a very important part in DNA replication. Helicase is an enzyme which is play an important part in DNA replication. It breaks down the bond which are present in between DNA, which is present in DNA strand. These strands are going to be breaks up or break down by the enzyme. This is called helicase enzyme. I am drawing here already the helicase enzyme. This enzyme is going to break the double bond, triple bond, which is present between the AT and GC, adenine and thymine, guanine, and cytosine, which are going to be broken down between the two bonds which are present between AT and GC are going to break down by the enzyme called helicase. So topoisomerase helicase followed by another enzyme. This is called, or you can say protein. This is called Ssb protein. So what is Ssb protein? This is a single stranded binding protein, or you can say Ssb protein. The single stranded binding protein or Ssb protein, which is going to be pulled the two strands to opposite direction. This strand is pulled by this direction and this strand is pulled by this direction. That's why the DNA strands are going to be separate and they cannot be wrapped around it. To prevent the wrapping or prevent the coiling of the two strands, this Ssb protein play very important role. It pull the two strands to opposite direction, and the helicase at the same time going to break down the bond which is present between AT and GC, adenine and thymine, guanine and cytosine. Now another enzyme, or you can say this is a short piece of RNA. It is called primase. Primase is nothing but a small or short piece of RNA which are added at the 3' hydroxyl group. 3' hydroxyl group. Short piece of RNA. Primase is a short piece of RNA which are helps to adding the short sequence of nucleotides at the 3' hydroxyl group. Then another enzyme that is called ligase. So ligase is a type of enzyme which helps to join or ligate the two small sequence of DNA. So there is enzyme, this is called ligase. So ligase acts as a joining unit between two nucleotides. So this is called ligase. So these are the enzyme, 5 types of important enzymes which are involved in DNA replication. These enzymes are helped to start, break, separate, and adding and joining activity are done by these types of enzymes. So I hope this video is helpful for the DNA replication. At last, we are going to discuss about its all our view about DNA replication. So in DNA replication, the DNA is going to be open up and it produces two DNA. Suppose a cell it contains the DNA. This is the DNA, and when it is open up, it produces two strands of DNA from one DNA. Suppose this is A, this is B, and suppose pDNA. So pDNA is going to be replicated or unwinding or open up by different types of enzyme activity, and it produces two DNA. That is the pDNA replicate to produce two DNA. That is one is DNA A and another is DNA B. So DNA replication helps to produce two DNA strands from a single parent DNA strand. From a single parent DNA strand, these two DNA are going to be produced. I hope this video is helpful to you about the DNA replication idea and the enzyme involved in DNA replication. In the next video, we are going to discuss about mechanism of DNA replication. If you like this video, please subscribe and share this video. Thank you.